We have a really loaded agenda tonight, so we'll start. I'm Thomas Duchesne. I'm a researcher here at the Goodman Cancer Research Center, and it's an honor for me to facilitate this seminar series, this public forum series. Why do we do this? It's really important to start from the start, and asking why do we do things. The reasons behind this public forum is because research, the cutting edge research, takes a long time before getting to public. And in the case of cancer research, this knowledge can be life-changing. We had quite a few events and incidents or cases where when we bring the latest in cancer research to an audience like you tonight, it can alter and change first how people, what people perceive about cancer as a disease, but also it enables and empowers patients in their decision-making and next to kin also in orienting treatments as well. That's one thing. So bringing science to people and enabling people that live with cancer. Directly or indirectly, now we're talking about 1 out of 2 people will, in their lifetime, be touched by cancer. And if you think about next to kin, then it's everyone who will actually have to deal with cancer from close or far. So might as well face it with knowledge, which is, in my view, the only and the best way to face it, that and hope. The other reason why we do this is because the GCRC, the Goodman Cancer Research Center, has been really well supported by you, by the community. It's a center that embeds in the Montreal community, but in the broader community in Canada and in the world, among scientists, among students, amongst trainees, amongst also donors, because we're grateful from the support that we had over the years. And it's our way to show a little bit of what we do in our labs. Science very often is hidden in silos somewhere in ivory towers. It's about time that we bring that science back to you in a wise way to thank you for your support as well. So that's why. That's me. Hi. And I've introduced myself, but I also need to mention the people who actually started this public forum, the facilitators before me. There's several decision makers behind this and several from the communities I want to salute. But at the same time, I need to thank also Michel Tremblay and Nicole Beauchemin, who have sailed this boat and kept it afloat and then beautifully over the years. I'm glad I'm actually now the coordinator and the co-facilitator for this since last year. Tonight's about trying to bring substance to what many of us start to perceive as what we call a buzzword. What is AI really? What does AI mean in concrete terms? You ask multiple people what the definition of AI is, and you'll find just probably as many definitions, right? So, but there's actually science behind it. There's great science done here in Montreal and in McGill, and we'll feature some of it tonight. But what's the intersect between AI now and cancer? Can we leverage the latest in computer vision, for example, in neural networks and big data analysis? Can we use this to accelerate discovery and accelerate ameliorate prognostics, and also make smarter decisions about treatment and anticipating the outcomes of treatment? This is what tonight is about. And we have a great lineup of speakers from McGill, from within McGill and sister institutes. The first speaker tonight will be Talar Bell. She's a professor at the Department of Electrical and Computer Engineering. She will present essentially the essence of computer vision and also its implications in tumor and stratification, and also how the latest in science can be used in diagnostic as well, medical diagnostics. Jan Sunjan and George Shinuda will, from the Gerald Brofman Department of Oncology, will present the incidence and the importance of AI and deep learning in head and neck cancer in optimizing treatment, detection and treatment. Then we'll have a presentation by my colleague here from the Goodman Cancer Research Center, Luke McCaffrey. He's going to present some of his recent projects in pattern analysis and tumor identification and stratification. And finally, we'll hear a great story from John Kildea and Tarek Hijal from, again, the Joel Bronfman Department of Oncology, when they'll present their development of the OPAL application. It's a beautiful story, and behind it is a profoundly human story. It was inspired by, as you'll hear, one of their colleagues who passed away recently. And it's a razor-sharp focus into getting data from patients that are willingly giving their data, enabling a better treatment, a better follow-up at treatment, and then using this data to mine it in order to improve the treatment afterward. So that's how we'll end the presentation session. What's going to happen afterwards is you'll have, during the evening, you probably have received a white piece of cardboard where you can write the questions that you have in mind, that pops in your mind at any moment during the evening. You write it down, and at that moment, at the end of presentations, there will be runners grabbing those pieces of white cardboards, bring them to me. Be careful to ask questions that relate on general terms, not personal diagnostic cases and stuff like that, so something that appeals to the audience as a whole. If you have more personal questions, you can come down in the end, and the speakers will usually hang out for a few minutes afterwards to connect. But try to ask questions that are general. We always get wonderful questions from the audience. We're always impressed at the quality and the excellence of the questions that are asked. So it's less scary than having to ask at the microphone. By all means, ask away. And then we'll go on for about maybe 20 minutes to half an hour with our full panel here in front answering your question in a direct conversation of back and forth. All right? So that's how it's going to unfold tonight. As you heard, I speak English, but if you are more comfortable asking the questions in French, if you want to speak out or ask a question in French, it's not a problem. I can translate anything also if someone asks as well during the evening. Je parle très bien français, puis vous êtes les bienvenus de faire n'importe quelle intervention en français aussi. C'est un plaisir de vous répondre en français as well. So on this, without further ado, I will introduce you to Tal Arbel. Tal is nothing but a leader here in McGill in AI. And do I have your presentation here? This one? Okay, that's fine. And I'm glad she's with us tonight because she pioneered a lot of the excellent science done in AI here in McGill. And
okay, thank you very much for the kind introduction and for allowing me to speak a little bit about my research. My name's Tal Arbel. I'm going to be speaking about machine learning in medical image analysis, and I'm going to talk about moving towards imaging-based precision medicine. And because I work quite a bit with brain imaging, I thought I'd introduce myself by showing an image of my brain. So the goal of my research program is really to bridge the gap between three very different fields, and I'll explain each one as we go. And it's the fields of computer vision, AI, which includes machine learning, and I'll talk about that, and medical image analysis. And I'm sort of at the intersection of these three fields, and the research that we do is all linked to sort of real clinical applications with impact in medicine. So the majority of the work up till now has been working on sort of neurology, neurosurgery, and neuroscience in general, but the methods are very general and applicable to a wide variety of domains. So again, the research is not possible without interdisciplinary collaboration. So I work a lot with machine learning researchers, biomedical engineers, clinicians, and I also work with sort of in the clinical domain and also with companies that work a lot in the medical industry. The main application areas that I've been working on are multiple sclerosis and brain tumors, but again, it's very general and there are a lot of potentials for other kinds of cancers. So what is computer vision? That's my main area of research. That's where I got my master's and PhD in this area. So computer vision is a field that involves developing these computer programs that automatically analyze and try to understand the content of digital images. So if you can see here, images can come from your camera, they can come from movies, they can come from sports, and again, they could be images or videos, it could be surveillance and medical imaging, it doesn't matter. We're trying to get sort of an area that's called artificial perception, getting the computer to understand what it's looking at through images. Now, machine learning is an area within the greater space of artificial intelligence. Machine learning involves developing programs that allow the computer to sort of automatically learn patterns of data. So in the context of computer vision, machine learning involves things like, if you look at this picture, a training phase where the computer gets a lot of images, for example, of dogs and cats, and it learns patterns that are distinctive of each. And then you get a new test image, for example, this dog picture, and the computer is asked, what is this? And the computer then spits out, based on what it learned, it spits out a label. This is a dog. Okay, so machine learning, in fact, has led to huge breakthroughs in the field of computer vision, really outperforming all the other methods that have come before it. And you've seen a lot of the application areas, face recognition, tracking, scene understanding, and it's led to a real economic boom for startups and, of course, a lot of the big companies that we're familiar with, like Facebook and Google and so on. And here's an example just depicting sort of autonomous driving, where there's a lot of work in machine learning and computer vision. Now, machine learning in medical imaging has the potential to make huge breakthroughs in medicine and in healthcare, helping with things like patient diagnosis. So here's an image showing a diagnosis of breast cancer. Understanding how diseases evolve. So for example, you have a patient that would, this is a normal brain, and then you may have a transition to mild cognitive impairment or Alzheimer's. And predicting patient outcomes from images. So it would be amazing to be able to look at a specific image and have the computer predict the patient outcomes. So for example, survival time and other issues. Also speeding up and making accurate clinical trials so we can develop new, much more effective treatments. Here's an example of a responder to treatment where you can see the tumor is shrinking over time, and permitting advances in personalized medicine. So this is like, for example, the computer is presented with an image of a patient, in this case a brain image, with a few lesions, and the computer predicts that there will be no change in the future. So the clinician can then say, all right, the computer thinks this patient will be stable, so I will not, for example, change or escalate the medication. So in this talk, I'll talk a little bit about some of the work that we're doing in machine learning for medical image analysis. I'll talk about some of the work that we're doing in detecting and segmenting lesions and tumors in specifically brain images of patients, and a little bit of work that we've done recently on conveying confidence back to the clinicians as far as the predictions of the computer. And I'll talk a little bit about the beginnings of the work that we're doing in precision medicine based on imaging, with the hopes of going more towards that direction and improving drug development. So one of the areas that I've been working on for years is brain tumor segmentation. And segmentation here, what we refer to is, you can see on the left, these are two different patients, and they both have brain tumors. And on the right, you can see what we mean by segmentation. It's basically delineating the tumor from the healthy tissue. And that is very important clinically in order to diagnose the patient and also to stage the patient in order to determine what kind of tumor or what kind of cancer they have, to predict outcomes, and also for surgical planning, so be able to determine how to properly resect the tumor. So in this case, you can see an example where there are a number of colors. Here, we're segmenting tumors into different substructures. So you can see, for example, in the pink is the necrotic core. The green around it is the edema, or the swelling around the tumor, and there are other areas of the tumor depicted. So you can see there are a lot of challenges in getting a computer to automatically segment the tumor and its constituent parts. Tumors can vary a lot in terms of size, in terms of shape, their position, in this case within the brain. And of course, the boundaries are very, very subtle. So here's some work that we've done in developing machine learning tools for brain tumor segmentation. Just show you a couple of results with high-grade and low-grade glioma cases. So here you can see these are two different MRI of the same patient on the left. And then it's the result of our algorithm right next to the ground truth manual segmentations that we have. So you can see that it looks very much like ground truth. Machine learning was able to do this quite well. And in fact, we had one of the top performing methods in these sort of challenges or competitions that we have in our field. The important things, of course, is clinical impact. So we have a very strong collaboration with a company called Synaptive Medical. Synaptive Medical is based in Toronto, and they build neurosurgical imaging platforms that are used by hospitals all throughout North America and really for surgical planning. So the important thing then, you can provide the surgeon with the segmented tumor and all of its constituent parts, and then the surgeon can then determine the best trajectory to resect the tumor during the operation. So here's a good example of machine learning methods leading to real clinical impact. Of course, it's not always common that you would have sort of one big tumor. There are many contexts within cancer and within a lot of other diseases where we have a lot of sort of unknown number of these focal pathologies. So here you can see some examples with breast cancer and others. And the idea here is that we want to find all of these focal pathologies, in this case cancerous lesions, in order to diagnose and in order to stage the disease. And so here there's two parts to the problem. First, we have to perform detection, and we need to find all of these sort of lesions, and then segmentation, delineate the boundary around them. So I've been working for years on a problem, a very similar problem within the context of multiple sclerosis, which I'll tell you about. It's a very similar problem in terms of the segmentation and detection, and where we've had some real clinical impact. So multiple sclerosis is the most common neurological disease affecting young people, and there currently is no cure. And one of the hallmarks of multiple sclerosis is the presence of these brain lesions which are visible on MRI. So you can see here sort of, this is T2 MRI, you can see these bright spots, and these red areas are depicting where the lesions are, the manual segmentation. So we have an industrial partner, actually here in Montreal, a spin-off from the Montreal Neurological Institute, which provides the analysis to the pharmaceutical industry for MS clinical trials for pretty much all the MS new therapies in the world. So they've provided us with what we call big data, which is tens of thousands of patient scans from different clinical trials, from different centers, and from different scanners. And we've worked on a number of different machine learning models in this context which are applicable directly to cancer. So that's why I'm bringing it up. So the first is the appearance of new or enlarging lesions, or for that matter, cancerous tumors, on a succession of MRI images are really important because they tell you something about new disease activity, and it's also very important because it tells you about how it allows you to monitor the treatment effect, both in the clinic and in terms of the clinical trials that people are running. So we developed the first machine learning method to detect and track the evolution of the lesions over sequential magnetic resonance images. Here you could see the baseline MRI and week 4, week 12, 20, and 24. So you can see the patient's image over time. And our algorithms have automatically detected not just the lesions over time, but differentiated stable lesions, which are seen in red, new and enlarging lesions, which can be seen in green, and blue is showing lesions that are shrinking over time. Another important marker of new disease activity can be found through contrast-enhanced imaging. This is used for many different diseases. It's used for multiple sclerosis. It's also used for cancer. So the idea here is that you have a patient and you inject the patient with a contrast agent, and in this case it's gadolinium, which is a common contrast agent, and you look at the image before and after contrast. So here you could see a brain image before and after contrast, and a portion, there's some enhancement that takes place due to the contrast enhancement. And you look at the enhancement, and there are enhancements in there that are indicative of new disease activity. And we'd like to find those areas and be able to detect them. They're very important because they tell us the disease is active, and they also tell us that, for example, if we have a lot of them, that, for example, the treatment's not working or this medication in clinical trials are not working. And so you can see here the result. These are the manual labels of what we're looking for. We're hunting for things that are very, very small. And there are other areas of the brain here which also enhance. So we have to differentiate the part of the enhancement that really has to do with the disease from other enhancements. So here's an example of very similar enhancement in the context of cancer. So again, we inject the patient with a contrast agent. You see a lot of bright areas, and we want to know among all the bright areas that we're looking at, are there new cancerous activities, and where are they, and how many elements are they? So here we developed the first machine learning method to actually detect gadolinium-enhanced MS lesions. I'll just show you 3 specific cases here. These are 3 different patients. And the first image is the original MRI, and the last image is after contrast enhancement. And in the middle, you could see everything bright is everything that enhanced in between the 2. So among all the things that enhance, we're looking for this tiny one little lesion, and our algorithm was able to find this one lesion in each one of these images, this gadolinium-enhanced lesion. So the important thing, again, these machine learning algorithms to detect and segment lesions were placed into the commercial software pipeline of our industrial partner. And so before, we had sort of very time-consuming and inconsistent manual or semi-manual segmentations by neuroradiologists, and these were replaced by much more efficient and much more consistent machine learning methods. So while that's good for the company, it definitely improved the efficiency and the precision of the analysis by 5-fold, saving them time and money. Much more importantly is that it improved the treatment analysis. So we have much faster treatment analysis for clinical trials. We can get drugs out to patients a lot faster, and we can stop trials that are not working faster. And so the segmentation and detection algorithms that we've developed in our lab are used for almost all the new multiple sclerosis drugs currently used in the world. But of course, machine learning can make mistakes. Okay, these are very hard problems, and we can miss a few lesions, we can have a few extra lesions, and so one of the elements of research that my group is looking at is sort of bridging the gap between machine learning and the clinic. So if an automatic segmentation has lesions, has errors, and we hand these errors to a clinician, this leads to mistrust in the system, and that leads to a reluctance in adopting machine learning tools into the clinical practice. So here you can see ground truth, and here you can see what the prediction is, and then the clinician would look at that and say, I can't trust this model. So we're developing and researching machine learning techniques to actually convey uncertainty to the clinicians, to convey the degree of confidence that the network or the model has in its assertions and predictions. And we've done this for lesions, we've done this for brain tumors. And the idea is that if you convey, and there's little question marks here, this, the uncertainty to the clinician can then look at the results and say, hey, the model's uncertain in certain areas, I'm going to review them. And so the idea is that uncertainty estimates will be part of the framework where machine learning can then be used to accelerate the clinical workflow rather than replacing them. So that's very important because our thoughts are uncertainty builds trust and faster review. So for example, in this case, the clinician will be able to determine which cases to review based on the output. And finally, I'll just talk a little bit, just for a few minutes, on some of the recent work that we're doing on automatically predicting future disease activity from images. Okay, so the idea is, and this is whether we're talking about lesions or tumors, the idea is to try to predict what will happen to this patient's image in the future, based only on imaging, so not other clinical parameters. So for example, if you look at the bottom, you will see a baseline image with a couple of lesions associated with it, and an inactive patient will have no change in that time period. And an active patient, you will see, for example, there's a new lesion, which we can see in blue, and we have a growth of a lesion, which we can see in yellow. So this is an active patient. So the ability to predict future activity or MRI disease activity based on only imaging is really important because it will help us understand the natural disease worsening. But much more importantly, it could help us have automatic risk assessment. So if the computer is saying that this person will have many new tumors in the future, then potentially the clinician can then select treatments that will be, let's say, more aggressive in that case and make informed decisions. And finally, we're also looking, of course, this will be helpful in predicting treatment efficacy, whether this treatment is working or not on a group of people, and also identification of potential treatment responders. So I'll just speak to that rather quickly. So we have developed the first machine learning models for predicting this future MRI lesion activity based on baseline. And because we have clinical trial data, we built different models for placebos for people, let's say, on treatment A and treatment B. And this permits us to actually automatically identify these potential responders to treatment. Because even in a failed trial, we know that it refers to the group, but there may be potential responders within that trial. And so the idea is, can we find them automatically through our machine learning algorithms? And then even in the failed trial, we can actually then try to determine how many people actually did respond. And so this leads to several possibilities for cancer care. The first is trial enrichment. And so here's an example where we have a patient image with a brain tumor, and we go back and we say, hey, we've seen people with this type of imaging before, and these type of imaging, for example, these types of images indicate that these patients had tumors that shrank, okay, with a specific clinical trial drug or treatment. So if the computer says that patients with an MRI like this one will have a tumor that shrinks, then we can do something like trial enrichment, which means that we can take the patients with similar MRI and bring them to clinical trials that are new and enrich them. And finally, where we're going in terms of our research is in terms of personalized medicine. So you have this case, for example, we have a patient that comes in with this brain image, and we have a model for treatment A, and we have a model for treatment B. And in treatment A, the model says that the tumor will grow. And in treatment B, the model says that the treatment will shrink. So this information can be provided to the clinician, and the clinician would therefore be able to say, I think treatment B would be a better option for you. So I just want to conclude with a positive note that we are on the cusp of another technological revolution, and the idea is AI in healthcare. So machine learning has enormous potential for impact in cancer in terms of precision medicine and drug discovery. And Montreal, there's a unique opportunity in Montreal to actually grow as a hub for research and innovation in this area. We have MILA, which is the Machine Learning Institute started by Yoshua Bengio, for which I'm an associate member. There's the Goodman Cancer Center, for which I'm an associate member as well, and other research centers with clinical, which are looking at other types of cancers, and people who are working on machine learning for computer vision, like those of us at the Center for Intelligent Machines who are part of electrical engineering and computer science and in other universities. So we have a very unique opportunity to make Montreal the hub of this type of research and to attract researchers and students across multiple disciplines to Montreal to grow this area. So I just wanted to show you a little bit. This is my team, past and present, and these are my sponsors, and I just wanted to thank you for your attention.
So good evening, everyone. We're going to continue along the same vein. So my name is Jan Seuntjes. I am a physicist working in medicine and healthcare. And we're going to talk about head and neck cancer radiotherapy and deep learning. And so I would like to start with an observation. And the observation is as follows. Humans can process only about 5 to 8 variables at any given time. And so in the medical context, people work in teams in an attempt to mitigate this. And so we collect information and experience from different expertises together in order to increase the number of variables that we can base a decision on. The other observation is that fewer than 3% of the patients participate in clinical trials. And clinical trials are the cornerstone of medical hypothesis testing. So, nonetheless, when a patient enters the hospital, there is a wealth of data being collected. And we can just look at the time the patient presents to be examined. We can look at medication, we can look at diagnosis, imaging, treatment plans, and so on. All different types of data that are being collected. And this data could be useful is useful to prescribe the treatment plan and to treat the patient. So let's look at, for example, one type of data is imaging data, and we've heard about imaging data in just the previous talk. Here is different type of imaging. The upper panel shows positron emission tomography data, and the lower panel shows computer tomography data. And they reveal different types of information about the patients. And so if we have methodologies to use all that data, then it would be fantastic to use computers to analyze these data. So right now, there is health professionals looking at these data, radiologists and radiation oncologists looking at these data for treatment planning and the like. If we could be using artificial intelligence technique which look in a different way to the data, things that we cannot see, then the data could be used in a more complete manner. So I would pose the following. The hospital is a big machine that generates real-world data. This is not clinical trial data. This is dirty data, but it's data. And we could use real-world data to help us predict, using AI, how the patient will do in the future, in a way that we have not done so far. And then in a further step, we could optimize an intervention based on learned experience from previous patients and combine this with data for a particular new patient. So I'm going to focus down a little bit on radiation oncology, which is the specialty I'm associated with. And we have different types of data that come into the scene, from diagnosis to pre-treatment to treatment. Diagnosis, we have imaging data, diagnostic imaging data, computer tomography, MRI, positron emission tomography, potentially biopsy. Pre-treatment data, which is associated with the simulation. Simulation means that we are bringing the patient in to simulate the treatment, and during that phase, there's also extra data. And then we have the actual treatment, where we deliver radiation in radiotherapy based on a treatment plan that we have created. And the type of imaging that is used, I already emphasized a few of them, can be X-ray based, which reveals anatomical information, or can be nuclear medicine based, which reveals more metabolic information, such as positron emission tomography or SPECT, single photon emission computer tomography. And then ultrasound is another type of imaging modality, or magnetic resonance imaging, which is extremely good in showing tissue contrast as well as functional information. And then the patient moves on and goes to the radiotherapy machine. We use all that data, and the patient goes on treatment. And on the right-hand side, you see a little movie showing that even on the treatment, we have imaging data being collected. And in this case, the imaging that is collected on the right-hand side is used for setup purposes, but it's not used, for example, to see how the patient is doing in terms of treatment control. On the left-hand side, we have the radiation delivery, and the radiation delivery, you can see the color showing up in the patient head there, which represents radiation dose. And that radiation dose is achieved by an optimization process. And that optimization process takes into account the dose prescription that we want the tumor to get, while respecting normal tissue constraints on organs that we want to spare. And that is shown slightly in the slide here, where we have tumor target volumes, we call them target volumes, where we optimize the dose to, and we try to avoid dose delivery to the organs at risk. So there's a number of entry points for AI to play a huge role in delineation. Dr. Tarbell already talked about delineation or segmentation, dose optimization, working on the dose optimization algorithms, and adaptation, because during the delivery the patient changes. And Dr. Shinura will come back to that point later. I will focus now on outcomes prediction, which is the area that Dr. Tarbell also already looked at. So I'm going to talk a little bit about what we call radiomics, and that's the use of texture information in the images. That's information that the radiologists cannot see, or radiation oncologists cannot see, but it's there. And it comes from the hypothesis that perhaps genomic heterogeneity of aggressive tumors translates into heterogeneous characteristic at the anatomical scale. So we have anatomical information through CT, and there is changes that are visible, and we can maybe pick them up using these textures. So imagine all this type of information that is shown in the little cartoon here. There is medical imaging information from which we potentially could lift textures, or there is pathology information, or even blood biomarkers. That's a different type of information, but it can also be used using machine learning. And there is even administrative information on the patient. So if we could use these textures to make predictions, that would potentially be useful for risk stratification in the same fashion as Dr. Tarbell described. So this is an example of the so-called GLCM texture, which is one type of texture that it's a mathematical concept, and it shows relations or the spatial distributions of patterns of gray level variation throughout the image. And we can compare patients that develop metastases with patients that did not develop metastases and analyze these textures. And we can come up with potentially features in the numbers of hundreds and several hundreds. And these can be used to correlate two outcomes that we have about patients in a training set. So the idea would then be in the radiomics world, where we use these features, these textures, that we take imaging information, we segment, we look at regions of interest, and we lift these features out of the image, we analyze them as in a big data fashion, and we build machine learning models that link presence, information in those features to certain outcomes. And when a new patient comes, we can hopefully use that machine learning model to predict or risk stratify the patient. So we have worked on this using with data from 4 hospitals in Montreal. 2 hospitals we used to train the models, and then 2 other hospitals we used to test the model. And the outcomes that we looked at were distant metastases, local regional failure, and overall survival. And so here's sort of a pictogram of what the analysis consists of. You have positron emission tomography images and CT images, and in the upper left corner. And then we have features that we extract from these different images. We built a machine learning model, in this case a random forest model, and then we come up with these risk categories in the upper right corner. In a later stage, we have learned, or we have been playing with deep learning model, which actually steps away from using explicit engineered texture models. And these deep learning models are essentially convolutional neural networks that were trained on CT images, on PET images individually. We then lock down the weights, and we also include clinical inputs. So here's a way to build a network that combines different types of data in one machine learning model, then uses convolutional layers to arrive at a score, a prediction score. And for example, we have shown that we can get an overall prediction accuracy which is much enhanced when we start combining different types of data. And this type of models is also much more prone or much more robust against missing data. And when we start using real-world data, which is available in a clinical environment, this is a distinct advantage, that the model is somewhat robust to missing data. So let's go back to the bigger picture then. So a patient comes in the hospital, gets CT imaging, different types of imaging. As a function of time, we can build we can use all that information that is collected, and that information could be images, like we discussed before, or could be doctor's notes on which we can perform natural language processing. And the idea is that we classify that information in different categories. And these categories would then be stored, and for each of these pieces of information, we would have a confidence interval about the accuracy of it. So we are working in a collaborative group called the Medomics collaboration to develop this kind of platform for hospitals to use. So it's an open source concept where we combine the data as a function of time from different systems, from different deaggregated systems in the hospital, and bring it together in this type of framework. And so the Medomics Consortium was in fact, the architect of it is in fact a Quebecer who is now in San Francisco, Olivier Morin, working together with Martin Vallière, a postdoc in my group, who came up with that idea. So to conclude, the theme of this evening was the future of AI in radiation medicine is now, or in medicine in general is now. AI can indeed play a major role in optimization and streamlining of processes, and medical decision support could be based on real-world data that we can collect from hospital now. In the particular radiation delivery, we could optimize it based on real outcomes instead of radiation dose models, and that will a field that is emerging, and that is called outcomes-based treatment planning. So it's very exciting to go in that direction, and we hope with Medomics that we can work in that direction. However, I have a few concerns as well. We are currently facing a siloed structure of unstructured data in different unfederated systems in the hospital. There is no standardization. There is a non-systematic recording of patient outcomes. The recording of patient outcomes has to be done by medical professionals, and so the use of this type of data hinges on the quality of that recording. We need a clear language, what is called ontologies and nomenclature, and maybe a very important point is that AI models that render medical decisions must be evaluable and interpretable. And Dr. Arbel already gave us some glimpse on work that is being done in her group. And I think that's extremely important because nobody will trust a computer if the computer is if we don't understand where the decision comes from. And then what is not addressed so far at all is the ethics framework that we will need for patient data and AI. And with this, I would like to acknowledge my group, which is on the left here, postdocs and students, and on the right-hand side, collaborators, clinical collaborators at the MUHC. Thank you very much.
Okay. Good evening, everybody. Bonsoir. It's my great pleasure to be here today to welcome all of you for coming to this forum, public forum that's very special for McGill University, and I'm proud to be one of the speakers tonight to welcome you. Thank you, Dr. Duchesne, for inviting me. It's a great honor. And tonight what we'll try to do is an overview of the radiation oncology involvement with patients with head and neck cancer, and how the artificial intelligence would have an impact and improve the precision of the process. The patient comes for head and neck cancer, and we have to gather a lot of information. We have to interview the patient, examine the past medical history. We have to confirm the diagnosis and the staging, and for this we use a lot of imaging, including CT scan, MRI, and PET scans. The patient has also to have a pathological evaluation of the lesion, and now it is not the objective to say whether the patient has cancer or not. It is the subclassification and the depth evaluation of the histological feature of any tumor. We're learning that no two tumors are comparable or similar at the genetic level and the physiological level. I started my training in radiation oncology in 1984, and I'm still learning. It is not an area where one can express or say, I know it all. It is very humbling, and every day there is new information that comes, and we need to be up to date in order to deliver the best treatment to our patients. So I also learned that if we don't do research, if we don't apply new knowledge and get ourselves into clinical and academic research, our practice is sterile. And at McGill, there is always this extra push for further research and further frontiers, and I'm proud to be part of this community. So after we reach the diagnosis and complete the staging, we present the patient to a tumor board where the patient benefits from the input of multiple specialists like surgeon, medical oncologist, radiation oncologist, pathologist, a lot of intervening medical caregiver who will give the best option for the patient. Is not only in one hospital, but we do it in teleconference, and we have multiple institutions converging their opinion and recommendation towards the patient, every single patient that we encounter. That's where we make recommendation and treatment option, and then we encourage all the time the patient to participate in clinical trials if they are available and if they are applicable to the patient. So the process of radiation treatment, I think Dr. Swanjis have presented most of the steps, and what I'm going to do today is, in these few words, is to show where artificial intelligence can have an impact. The process is to stabilize the patient, to have an immobilization, to do the best imaging possible, to fuse it with the scan in order to delineate the organs that we need to protect, organ at risk, and to identify the tumor volume that needs to be treated to the maximum safe dose that we can achieve. And for this, imaging is essential. We take the patient, we have a CT scan for doing the simulation, but recently we are able to do an MRI that will be done at the same time. And I think you can see the importance of the better delineation with a different modality instead of depending only on CT scan. We can do fusion with PET scan. We can do fusion with multiple imaging in order to help the identification of the organs. And this will help us to delineate the organ, and this can be done by auto-segmentation, and this will save a lot of time for the people who have to go through slice by slice and contour the organ at risk and the tumor that to be treated. And the head and neck area is 20 centimeter or plus, and we do thickness slices at 3 millimeter. Every 3 millimeter there is a slice, and on each slice we have to contour multiple organs. It can go up to 25 organs. So you can understand the process is very time consuming, and it is a precise process, needs to be done carefully. So once these organs are contoured, we assign a dose to each organ, and we decide on the amount of radiation treatment that is required for each organ that needs to be spared based on clinical information from before, and the dose that can be given to the tumor in order to achieve local control and cure for the patient. So this important area of dose calculation can be used as a indicator that can be used in artificial intelligence. And with more patients to be treated and more data to be entered, the machine can learn and give us the best combination of those to the normal tissue that can be spared and those to the tumor that has to be treated in a safe way and comprehensive way. So the artificial intelligence has a great potential to help us in improving the precision and the accuracy of the treatment so we can get better control with less side effects. So this is the basis of medicine, good outcome, and less side effects, the therapeutic ratio. So this is an area where we can benefit significantly when we apply it. So after that, we look at the dose distribution, like presented before, in the tissues and in the tumors, and decide the dose of radiation that is safe to be delivered. And this can be presented on what we call dose volume histograms, where each organ has a certain volume receiving a certain dose, and you can see the complexity of this analysis and how long it takes to be done properly. So here, artificial intelligence can, looking at multiple patients and thousands of patients, learn what is the better outcome and how to predict it from what we are doing. If it's not acceptable to the calculation or the prediction, we need to go back and do better. And in order to be able to predict outcome, the outcome has to be gathered or collected in a very clear, precise way. Otherwise, the information we enter in the computer will not have an equivalent in the impact of the treatment on the patient. So I have to say patient has a grade 1 toxicity of the skin, grade 2 toxicity of the salivary gland. If I don't quantify the outcome of the treatment, the use of imaging and artificial intelligence will not be of any value. So we have to make sure that when we say artificial intelligence, we have also to say, like we said, nomenclature and ontology, in the sense that we need to speak the same language, and this is happening. And I think in our department, our medical physics colleagues have done a great job by unifying the whole nomenclature in our department at the same level as all North America, so we can interact with other departments in an academic and meaningful way. So the requirements for a successful artificial intelligence is that we have to include all patients with different ethnicity, racial background, because it is not only the image, it's the patient behind the image. And we need to integrate all of this information and not to focus only on an image which can tell a lot, but I don't think, and I'm saying I don't think, I'm not sure, I'm not an expert, that it is not the whole story. There is more personal, individual data that we will learn about. I think genetic and molecular biology will have an impact in addition to the imaging, which is, I think, the most accurate information that we can get in an objective way nowadays. So this is the summary of the requirements, and we have to be very careful about applying the result of the artificial intelligence results before verifying them, before validating them and making sure that they are applicable and safe to apply to the patient. It's our medico-legal responsibility that we apply them in a careful way, not to be a hindrance to progress, but to do it carefully. And I think that's a message I would like to leave you with tonight. This is a new friend. We have to shake hands. We have to work together. And it is mutual respect, and we need to make sure that we use this information in the best to our patient. And to be careful that if we give wrong information, we will get wrong results. And this is a word from Charles Dickens. I thought it was pertinent. Always in time of revolution in anything in society or in science, we have to think about that this is the best of time and this is the worst of time. It all depends on what we do with it and how we do it. Thank you for your attention.
Okay, thanks. I'm happy to be here talking about some of the research that I've been involved with. Some of the project I'm going to talk about is a little bit different. We've heard about medical imaging, so MRI and CT. I'm a cell biologist, so I'm going to talk about a different type of imaging. So this is more pathology-based imaging, where you're looking at cellular and tissue structure. And so this is an image of breast cancer. So in my lab, this is what we study. We study breast cancer. And so what you can see here is that there's a bit of heterogeneity within these. So each of these green circles is a cell. Can you see the arrow? Yeah. So each of these green circles is a cell, and the blue is staining the nuclei. And then we have a number of different markers here that are showing in red and whatnot. What I'd really like to show is that there's some heterogeneity here. So you see there's cells that are small and some cells that are large. There's the tissue's organized in a certain sort of way. We see that there's a structure down here through the middle. And so a lot of these structures and a lot of the shapes and cells and patterns that we see here, this is what pathologists use to try and identify cancers and to assign a stage or a grade to them, and also to determine things like prognosis. So this image is done in our research laboratory. So it's done with a specialized microscope that has 4 lasers. In the clinic, it's a bit of a different process that they use to look at the images, and so this would be something that's something that we call histology. Okay, so this is really study of the anatomy of cells and tissues, okay? And so this is used by pathologists for a number of reasons. As I said, things like determining the cancer subtype, determining things like the stage or the grade, and also determining things like surgical margins. So after surgery, the tumor is removed, and then it's sent to pathology to determine, did we get the whole thing, or is it too close to the margin that maybe there's some left back in there? If that's the case, then the patient would maybe go back for surgery to correct those margins in regions that are more suitable. And so based on the information that the pathologist gets from these sections, from these images, then they can present that information to an oncology team, and then they're able to better decide on things like treatment options and treatment potential. And so this is essentially what it looks like here. We have 2 different slides. Its tissue is taken out, it's embedded in a wax, and then it's cut into very thin sections, so usually 3 microns. Okay, so this is millionths of a meter thin. And then these sections are stained, so this is where we get this kind of purple color here. So they're stained as a kind of a contrast enhancement, so you can see the difference in the cellular structure and the texture within the cells and within the tissue. So one of the questions we have is, how can we use technology and how can we use artificial intelligence or computer vision machine learning to improve some of this pathology? Okay? So with the digitization of histological samples, we now have the ability to apply things like computer vision and machine learning to these images. So typically these would be cut and they'd be sent to a pathologist who just looks at them under the microscope, makes some notes on the charts, and then that's it. So now we're moving more towards documenting these images, having digital representations of that. And as that grows, we're able to now to access some of that information for automation. So there's a number of benefits that we can envision from having this automated pathology or automated histology. And so as some of my colleagues have noted, the purpose here isn't to replace some of the clinicians or radiologists or pathologists that are studying it, but really to enhance their abilities, to improve the way that this is going to be delivered to the patient. And so one, I think, of the major improvements that could come from this is improving things like making the measurements more quantitative. Okay, so we know from breast cancers that the tumors are quite heterogeneous. So for example, an ER-positive breast cancer only needs 1% of the cells to express that estrogen receptor. Okay, so that means 99% of the cells are negative. Okay, so there's a tremendous amount of heterogeneity in there, and that's not always captured accurately by typical pathology reports. So we have the ability now to be able to very accurately measure some of these different parameters. Okay. The second is to standardize some of the analysis, to reduce inter-observer variability. So in cancers like breast cancer, it progresses through a number of stages. So there's a number of early precursor stages that aren't actually cancerous, and they're not really a risk to the patient in themselves. But they do have the potential to progress into an invasive cancer and become a problem. The challenge is that there's well-defined stages, but there's also intermediates or borderline cases. And so in these cases, depending on the features, one pathologist may call it something more advanced than another pathologist because it's in this gray zone on where to call it. So the challenge there is that in some cases, what it's being labeled as actually determines what the treatment is going to be. So I think, having these standards in there could improve the way that we're treating patients. And the last part is, we heard in the last talk, being able to increase the throughput. So like some of those medical images where they're manually annotating areas, histological images, if you think you have something maybe a centimeter thick and you're cutting it into 3 microns, that's an enormous amount of data. And so there's no way a pathologist could go through all of that. And so what they do is they pick representative areas and then they look for what the most advanced stage is. So do they detect an invasive cancer? And if it's yes, then they go. If they don't, they maybe want to section more, but it's just not feasible to look through all of those samples to try and find invasive cancer. So this is a way in which we can maybe increase those capabilities through image analysis. And so the short-term goals here are really to enhance the ability of pathologists to do their job, to improve some of the decision-making towards patient care. But a more long-term goal, I think, would be to develop quantitative measures that we could then compile into databases and use that information for better prediction. Okay, so we could apply that to future AI type of approaches where we can integrate a lot of these quantitative measurements into better approaches. So I'll just go through a little bit of some of the mechanics and some of the machine learning that has been utilized. And so one of the models, and we heard a little bit about it in the last talk, is about deep learning, or deep convolutional neural networks, okay? And so the idea here is that you would have an image. In this case, it's a bird. I think it's a crow. And so the image needs to be labeled. So in this case, you just circle what the image is, and you circle this and say, okay, it's a bird, okay? And then what happens is that image goes into a number of different layers. So the yellowy circles here represent the panel of images that are going to be loaded into the training system. Then these pink and green areas are this is kind of the neural network or the main basis of it. And so each of those different elements or components are going to look at different features within the image. So it might look at the hue, it might look at edges, it might look at blurriness. So there's a number of different features that it looks like, and it passes it along to each different section. So that's where it's called deep learning, because you have this deep sort of rich network of different analytical processes that are going on within the image. And then at the end, the orange ones is the output. So this is going to be the decision. And so the idea is that you can train the image or you can train the computer with a series of images. And then when you show it an image of something that's unknown, it's able to correctly verify or correctly identify that image. And so some of these models, these deep learning models, are often data heavy. Okay? So this means that you need tens of thousands of images, or at least many thousands of images, to get these accurate models. Okay? And so one of the questions could be, this works well for maybe a bird or a bicycle, but what do breast cancers and other solid tumors actually look like? And so here's an example. So this is a breast cancer. It's very solid. You can see it's dark purple. It's got fairly distinct margins, okay? If we look at it, here's another case of a breast cancer. And so in this case, you see there's a lot of pink. So this is protein deposits within the sample. The structure is quite different, and you can see there's a lot of normal tissue that's more this kind of white structure. Here's another example of a breast cancer where you have smaller lesions that are kind of evenly distributed throughout the tissue, okay? And then a final example here where it's mostly normal. You might not be able to see it, but there's a little area here that's actually got some of these dark purple cells. And so this is another region of the tumor. Okay, so there's a number of challenges here, right? We have, in addition to what we saw with the medical images, we have here, there's huge variability in what these things look like. And there's also differences in the colors. So not just that you have different proteins and things expressed that are being picked up differently, but you can have differences just based on the camera, how the histology was prepared. And so you have a huge variability in just what these images look like. So these need to be changed. And so it turns out that a lot of the great methods that have been developed for for medical imaging don't directly apply to what we're trying to do. So we've had to kind of go back and reanalyze and look at some alternative methods. So there's a number of challenges with machine learning and histological images. So one is that despite the prevalence of cancer, there's not huge available data sets for training. So we're not looking at data sets that have thousands or hundreds of thousands of images. We're really down to looking at data sets that maybe have less than 100 or slightly more than 100. So this is something that I think we need to improve in the future and to be able to build these better data sets. Another challenge is that digital whole slide imaging isn't widespread. So most hospitals do have things like MRI and CT for medical imaging, but whole slide imaging isn't widely available. So the machines are expensive, but also the data is expensive. So a typical image would have billions of pixels. Okay, so this is going to be big gigabytes of data for an individual image. For comparison, you might have hundreds of thousands of pixels in a medical image. Okay, so your orders of magnitude different on just how you analyze it. These huge data or these huge images also, the computational power, it can't analyze the whole thing. So you need to somehow be able to divide it into smaller regions. And then the last example is that the manual annotation is difficult. So here's an example of this solid tumor I showed you. And so segmenting this manually isn't too bad for a training set. But if you have a tumor where there's a lot of different regions and small little foci, we heard this in some of the other talks, that this is a major challenge in trying to identify and segment some of these smaller regions. And so this is my attempt at segmenting it. You can see that I maybe missed a few sections, but this would really, be something that would be overwhelming in a time commitment to try and generate large data sets for training where you could manually segment these. So one of the approaches we took is something that's called weakly supervised learning. Okay, so weakly supervised learning takes it requires a limited amount of information for the training set. So in a supervised learning example here, you would say, okay, there's a crow in the image, and then you would circle where that crow is. In a weakly supervised learning example, you might just say there's a bird or there's a crow in the image, and then you're going to let the algorithm try and figure it out. And so that's how these differ. So in addition to having a neural network there's other elements that enable focusing of the algorithm onto certain areas, and then you can go on towards pixel level annotation of some of those areas. Okay? And so there's really two elements that we wanted to get from this weakly supervised learning. Okay, so the first task would be image classification. So can we actually go and find, is there cancer in the slide or not? Okay, so this is actually a fairly major challenge. There's been a number of competitions. As we heard earlier, the machine learning community loves these competitions. And they're actually pretty cool. In my field, we don't really have this sort of thing. So it's really cool that there's communities that go together and they try to come up with the best algorithms. So the first challenge, that's been out there is image classification. So from diverse tumor types, can we actually be able to detect it? And the second challenge is going to a pixel-wise location within the image. And so this is just an example of what this might look like. You could have normal, you could have cancer. So it doesn't have to be binary. You could have a whole number of different cases in there. You could include things like benign. You could have different molecular subtypes. So any way that you can maybe observe a difference, you can add as a label and add that to the training module. And so what we did is we evaluated a number of different existing methods, weakly supervised methods, for their ability to actually detect whether, yes or no, there was a tumor in the slide. Okay? So at the bottom here in red, I'm just showing a supervised method. So this is kind of our theoretical maximum that we think we could achieve with these types of methods. And so that gives us 100%. And we were able to get between 99 and 100% accuracy with these weakly labeled methods. Okay, so that addresses the first part, and the second part is, can we get a pixelized localization? So some of these deep neural networks, it's a bit of a black box. So those multiple layers that it goes through, we don't necessarily know what's going on in there, and we don't necessarily know what it's making a decision on, okay? So we can't really access that. And so one of the ideas that we had is this idea of interpretability. So this is where you could actually access, where you get information back from the algorithm on where it's actually focusing. Okay, so we applied some of these methods to try and find out where these weakly supervised methods are actually looking within the images. So if we go back to a bird example, this is a sparrow or some kind of small bird. So we have the original image here, and this is one that we manually segmented. So this would be considered the ground truth, and so we colored that in red. And so if we look at these methods that had virtually 100% accuracy in saying yes or no, there's a cancer there, we look at what they're actually looking at for that information, and we see that to our human minds, it doesn't seem logical. So in one case, it's determining that there's a bird there, but the information it's gathering that from is actually in the whole image, or most of the image. And here's another image where it's actually ignoring the bird and it's looking at a twig. Okay? So there's a bit of a gap here on how these algorithms are working, what they're using to extract this information. And so what we did is we went back and developed our own methods to look at this, to enhance some of these weakly supervised methods, to try and see if we could get a better method for the image segmentation. Oops. And so what we did is we find that, yes, indeed, we can. It's not perfect, but I think it's a much improvement where we can see that most of the bird is now the object of that's being highlighted within these images. So birds are great, and so we asked, can we do this on histology samples? And so this is an example of a cancer, and again we have the original image and then we have our manually segmented one. We fed a number of images into the learning algorithm and we went through the whole process. So again, the current state-of-the-art methods that we see aren't doing a great job of segmenting. They're really looking at the whole image, and then the method that we have is a bit better, quite a bit better here. We can see that it's actually segmenting this tumor part here. So can we only look at advanced or more advanced tumors? We can also go towards So we can also go towards looking at benign tumors, okay? So we did the same thing. We've looked at benign tumors. We looked at breast cancers. We looked at colon cancers. And the algorithm seems to hold up in those different ones. So one of the things is you can't train on one type of cancer and then identify other types of cancer. So you really have to train each particular type of cancer. But we do find that the methods that we can use can be applied to different situations. So based on this information that we can segment on advanced tumors and more benign regions, we asked whether we can do this in a single image and whether we can segment different areas. And so we applied this to breast cancer samples. And again, what you can see is that we're able to pick up benign regions. We're able to get in situ. So these are malignant cells that are contained within the ductal structure. And we can also find invasive cancer structures within these images. And so to do this, we have a number of different processes. There's color normalization. So because each of the slides can be, different values of purples and pinks, we have to normalize these images. So there's a number of different pre-processing steps that have to go on, but it at least provides a proof of concept that we're able to achieve this. And so I'd like to talk a little bit now in the next couple of slides about where we plan to go with this and where our original idea was for predicting cancer in patients. So predicting in patients with early lesions whether it's going to become invasive. And it turned out at the time that we needed to redevelop a number of these systems for looking at these whole slide images. So I'll talk a little bit about ductal carcinoma in situ. So DCIS, ductal carcinoma in situ, is considered a non-obligate precursor of invasive cancer. Okay, so invasive cancer is what's lethal. So the cells are invading out of the ducts and they're getting into, they can then metastasize to other parts of the body. So DCIS is localized. So the cells don't leave the confines of the duct. And so this is not actually a risk to the patient. If it maintains its structure within the duct, it's actually not a risk. It's once it invades out. It's estimated, so when I say non-obligate precursor, that means that all invasive cancers are coming from a DCIS, but not all DCIS are going to become an invasive cancer. So it's thought that maybe 25 to 50% of DCIS will become an invasive cancer. So currently about 20% of diagnoses are of DCIS. So from the medical imaging that we saw, so mammography, those little white dots are microcalcifications. And so those are often associated with DCIS. And so that's one method that you can start to predict the presence of these. But it really requires going to this histology and these methods to fine-tune and look at whether any of those cells are actually invasive and breaching that membrane, in which case you would upgrade it to call it an invasive cancer. So the biggest challenge currently is that there's actually no way to predict whether a DCIS will progress. So DCIS have mutations that already exist in advanced cancers. So there's work looking at how the microenvironment is maybe able to, so that the cell and the tissue around the DCIS, how that maybe influences, but there's currently no method to discern those. So this is in my lab a major research goal, is to identify methods to maybe predict DCIS that will progress. And so what we want to do is apply some of these methods that we've established for identifying different types of lesions into DCIS lesions. And so then the idea would be that we could segment and determine whether, even though the DCIS is confined, are there cells in there that maybe have properties of an invasive cancer. Okay, so the idea would be looking for invasive cells in a non-invasive tissue. Okay, and so the idea is that this would then maybe predict DCIS that are going to progress. Okay. And so the idea would be, if we can identify these subregions, then as a basic scientist, it gives me the ability now to identify those. So what we would want to do is look for biomarkers that are maybe could be readily used to stain, and to understand the mechanisms of how that's going, and develop better biomarkers to predict this. And so just as a proof of concept, you know, we've gone through some of this, and we can detect DCIS where we're picking up regions that have invasive cells. And so this led us to ask, are there populations of cells within DCIS that do have invasive properties? And so this is just an image here showing a stain with particular markers. And this marker we only detect in invasive breast cancers. So we ask, can we also detect it in some DCIS? So in this particular patient, they had invasive cancer as well as DCIS. If we look at the DCIS, we can see that there are markers. So we do see that this marker is present in some of the cells within the DCIS. So we think that this is maybe a population that is more amenable to progressing. Okay? So here we have a subpopulation in the invasive case, it's all of the cells. And so we ask, can we actually go back even further? Can we go back into earlier stages? So here we're looking at now in the margins of the patient where it would be considered normal. So this is the normal adjacent tissue. So it's phenotypically normal. But right here you can actually see a single cell that's expressing this marker that's showing up pink. Okay? So we do think that these cells pre-exist at earlier stages. And so now what we're trying to develop is the machine learning approaches that will allow us to, in a high throughput way, try to identify some of these DCIS that maybe have features of invasive cancers, but they haven't yet progressed. So my lab is highly interdisciplinary. We have a lot of collaborations with different groups. This particular project on artificial intelligence is in collaboration with a group at ETS. So it's led by Eric Granger and Ismail Ben Aid. And so we're co-supervising some students and postdoc on this project. So Jerome Roney and Soufiane Belardi are the two who really have driven the projects and done the programming and the analysis. And I like to call them the real intelligence behind the artificial intelligence. So also I'd just like to acknowledge some of the funding. We have quite a bit of breast cancer funding, which is funding this project, but also a number of other projects that we have in the lab. Thanks.
Hi, everyone. My name is John Kildea. I'm going to start off this next presentation, which I'll be giving with my colleague, Dr. Tariq Hijal. And this is going to be a presentation about a mobile phone application. This application is called OPAL, and its goal, or our goal with this project, is both to empower patients and to inform research, both of them using this application. But it's also about the realization that this statement is very true. This statement that patients are the most underutilized resource in healthcare, and that as a healthcare system, we haven't been doing enough work in partnering with patients. And this, the work that we've been doing is what we believe we should be doing, is working with patients to improve the healthcare system. And as Thomas said, this is also a story. And like any good story, it has a beginning. So we'll start at the beginning. This started in 2014 when Laurie Hendren. Laurie was a professor of computer science here at McGill. She was a breast cancer patient beginning in 2014, and she was a breast cancer patient under the care of Dr. Tariq Hijal, who will be speaking in a few minutes at the McGill University Health Centre. And Tariq and I'm a medical physicist. We were already working together on various computational projects in radiation oncology and medical physics. And so Tariq, on having a conversation with Laurie, introduced her to me, and the three of us had a conversation about how we could work together to apply computational techniques in radiation oncology and in medicine generally. And the three of us worked together for 5 years as equal co-leads on this team. Equal co-leadership is very important because that meant that we had a patient equally co-leading the team with us at the very center and sitting at the table all the time and making sure that this project was going in the right direction. What we formed was what we called the OPAL Health Informatics Group, and that was a research collaboration to use data and artificial intelligence to improve the experience and outcomes of patients. And so when we were sitting together back in the summer of 2014, we were wondering what we could do together, and so we asked Laurie. We said, Laurie, you're a patient. What do you think we can do to improve the experiences and the outcomes of patients in the healthcare system? It only took her a second to say what she thought we could improve. And number one was she thought we could improve the pain of waiting. Now, in healthcare, we're always trying to reduce the pain of our patients. We're trying to make their lives less painful. What we didn't realize is that waiting in the healthcare system is also a pain. And Laurie really pointed that out for us, the pain of waiting. And the second thing was give patients their data. Laurie was a professor of computer science. She worked with data all the time. She came to the hospital, the healthcare system. She was not able to access her own medical data. And this for her was a huge problem, as it is for many patients. And as we went along, we learned the power of sharing data with patients. And you can see this just in this single example where Laurie, was her treatment plan in radiation therapy. Just by showing her plan, she knew which part of her skin to cover with sunscreen when she was out in the sun. And she was able to show her physiotherapist outside of the hospital which muscles were being affected by the radiation. So this is how we got started, working together with Laurie. We had these two things that we wanted to work on, and so we wondered how we could do it. We didn't think we could improve number two, but we thought we could work on number one. Number one being the pain of waiting. And Laurie's vision, I should say before I move on, Laurie's vision was that waiting will be less painful and that all citizens in Quebec will have full access to their healthcare data. And these are the two things that she identified and that we've been trying to work on. And Laurie unfortunately passed away at the end of May, just a few days after our group was awarded the Prix d'Excellence from the Ministry of Health. Laurie unfortunately didn't survive to see that, but our team was really grateful that the ministry recognized it and recognized that this type of work is important. And there was a nice article written in La Presse newspaper on June the 1st about this project and about Laurie's vision. So we'll go ahead and speak about how we are trying to achieve that. So number one was the pain of waiting. What we did is we got together and thought about how we could work on this, and we built a waiting room management system at the Cedars Cancer Center. So we built a check-in system for patients to check in with their Medicare card with a kiosk. We built a virtual waiting room so that the staff can see which patients are checked in for them, how long they've been waiting, and a system to call in patients with TV screens. And as researchers, this provided us with timestamp data, because now we could see when our patients were checking in, when they were getting called in, and how long they were spending in the examination rooms. So that was nice, because that provided us with data, and with data, as we've seen today, we can do artificial intelligence and we can apply machine learning. So we did machine learning to make predictions for patients about how long they would have to wait for their appointments. And this is the type of work that we were working on, but we wanted to provide these predictions to patients. And we started thinking about how could we communicate these. And we came up with a mobile phone app. So the idea was that we would make that patients would arrive at the hospital, they check in on their phone, don't need to go to the waiting room because the GPS will allow us to see that they're at the hospital. They can wait in the cafeteria, they can go to the bathroom, and when it's time to call them, they will get buzzed on their phone to come for their appointments. And it would allow us also to communicate these waiting times. Now, I can tell you the waiting time part is still a work in progress. It's not a problem that has been solved. It's a problem that we're still working on. But we realized when we were building this mobile phone app that now we had a way of tackling problem number 2, which is communicating with patients and sharing their data with them. And that's what we ultimately started doing. We kind of pivoted the project away from the waiting times, although it's still important, but now providing patients with access to their data. And this is what we built, Opal. It's a mobile phone app that empowers patients with access to their data, ultimately from across the healthcare system, initially at the McGill University Health Centre, and we're trying to embed it into care, embedded into the healthcare system, both for clinical care and for research. And Dr. Hijal will now give you a little tour of what Opal is and what it does.
Thank you. So what does Opal look like? You can see a screenshot on the left-hand side. It's, first of all, a personal health dashboard. We try to give all the information patients need at their fingertips, including appointments and appointment reminders. So when patients arrive to the hospital, they can see that they can check in for their appointment, as John said, GPS enabled. And Opal is also a full patient portal with access to their medical records for patients with all the data explained. For example, patients can prepare for appointments because they know when their next appointments are, and they're actually reminded with notifications of their appointments. When they click on an appointment, they can actually see, for example, the map. And when they click on About this appointment, they can have more information of what is the appointment about and how to prepare for the appointment. For example, you need to fast 4 hours before a CT with an IV contrast. Patients can also access their lab results. Here you can see the tumor markers that go down when chemotherapy is given and then go up when the tumor becomes resistant to chemo. So we're empowering patients through knowledge, but also through understanding, because every piece of data we provide to the patients, we provide information. So what does a tumor marker mean and why is it done by the doctor? There's also questionnaires that you can see here. So when patients check in and are waiting to see the doctor, we can send them questionnaires about their symptoms so that when they see the doctor has already looked at the answers and can focus the visit on where the problems are, rather than repeating questions where they can see that the patient is doing well. And finally, there's a lot of educational material provided. So this is automatically personalized to the type of disease and the type of treatment, and just in time when it is needed. We don't want to overwhelm patients with too much information too early, but at every step of the way, we give them the relevant information. So it is all integrated in the clinical workflow, as I just said. When patients check in, they get the questionnaires, they answer them, they see the doctor, they discuss the problem, and then they check out and go back home. How has the project advanced? So it's mainly over the past 6 months that we've been enrolling patients, and the curve shows that we're now over 500 patients using OPAL at the Cedars Cancer Center of McGill University Health Center. We also have a project where we're going to screen for distress in cancer patients at 6 other hospitals in the Montreal area, and that was done with the Ministry and the Canadian Partnership Against Cancer. Feedback from patients has been good. That's an example of one of our patients using the app. She found that she couldn't function without it because she knew as soon as she did a blood test whether her blood counts were correct to receive the next chemo, or whether when she would go home she would have to be worried about getting an infection because of low blood counts or not. And we surveyed patients, and 94% of patients feel that the app has improved their cancer experience. Everybody would recommend it to another patient. And we asked the question because there was fear from the treating team that giving all that information to patients would make them more anxious. And what we actually saw is that 60% of patients felt that getting their blood tests made them less anxious, and 40% it didn't change their anxiety level. And no patient actually answered that their anxiety was increased. So where are we going from here? I'll let John finish with the last couple of slides.
So thank you, Tarek. So the work is ongoing. This story had a start in 2014, but it is still going on. Right now, we're trying to develop a caregiver enhancement to this, and the idea is that patients should be able to share their data with their caregivers. So, for example, if we take the example of The Simpsons, Marge could be a patient in the hospital herself, but she's also the parent of Bart and the neighbor of Ned. And as Bart's mother, she should be able to see Bart's data. And as the neighbor of Ned, and she gives him a ride to the hospital, he shares his appointment schedule with her, so she's able to see his appointment schedule. So this is the next phase of the project that we're working on. But we'd like to go even further than that. We'd like to use AI to match patients with previous patients. Because as a patient, as you live through the experience of going through the healthcare system, you become an expert on it. And sometimes you're able to provide insight to other patients who might come along. So the idea is that we should be able to allow patients to connect with other patients who wish to volunteer to provide that insight. Or if patients leave behind written advice for future patients, we should be able to match them with that advice. We would like to bring this to another step, which is data donation. And this is really bringing the patient into the research. So by giving patients access to their data, they can donate it. And this is quite different than consent. We can consent, but we can't donate what we don't have. So if we're giving patients access to their data, then they can donate it. And potentially, if patients have access to their data from across the healthcare system, they can donate all of their data. And as researchers, that can save us a lot of time, because otherwise we need to go around to each of those hospitals and get those data to do the research. So getting them from the patient directly could save a lot of time. And also we can engage the patient, because we know that if you're going to donate to a charity, you like to know where your money is going. So if you're going to donate your data, you'd like to know what we're doing with your data. It's a two-way street. So we can provide information back. How are we using the data? We can provide newsletters about the research, etc. So there's the potential to bring the patient directly into research using this. So as we're moving forward, Laurie's vision is really our vision, and we're working with the Quebec government to try and get this out across the province so that we can really achieve Laurie's vision, which is that patients across the province should have full access to their healthcare data. And that's it. These are our funding partners. And really a big thank you to all the patients. Multiple patients, as well as Laurie, have participated in this. Multiple students at McGill in computer science and in medical physics, and as well as all the clinicians and the various stakeholders at the hospital that have supported us from the start. Thank you.
Wonderful. So this is the period of the evening where you share your wisdom, concerns, and questions openly with our panel member. I will call the panelists in front here, come and sit in front at the table, all the presenters. And to the runners back there, carefully recover those white cardboards. Please be careful, it's steep. There is an history of mishaps there. And while we recover the questions for the evening, I would like to mention that the next forum is on November 20th, and it's going to be on human papillomavirus. And I will just leave it here. So that's the next lecture here, okay? 2 questions. Thank you. The microphone is back. Already great questions, I can see. All right. Okay, so we have global questions, we have general questions, we have very precise questions. I will ask with one simple one, right? Who owns the data from a patient? Who owns it? And if the patients own it, why can't they freely have access to it? Simple question, but difficult to answer, I guess. Who owns the data generated from someone's clinical record?
So when we started trying to allow patients to access their data, we got a lot of feedback from the clinical teams that patients can't access their data without somebody from the clinical team looking over them. And we asked at medical records, and they told us yes. If you go to medical records and you want to access your data on a paper chart, somebody will stand over you and watch you do that. It turns out that that's been in existence for some time, and that's because originally charts were on paper. And so if you go there, they didn't want you to write on it or to change it, and they would look over you to make sure that the data was the integrity of the data was correct. So now that we're in an electronic era, it's a little different. So the data, I think, belongs to both the patient and the healthcare system. The healthcare system is the custodian of the data, but the real data belongs to the patient.
Clear answer. Thank you. We'll start with the questions from the audience now. So how can a cancer patient get access to OPAL?
So right now the project is running at the MUHC. So patients have to be patients of the Cedars Cancer Center. At the S1 level in the Cedars Cancer Center, there's the kiosk of the foundation, and they can just go and register for OPAL.
Okay. Considering that data plays a key role in AI, what has been done to accelerate and facilitate capturing, sharing, and labeling of data? I would say we're more capturing and sharing, right? What has been done to accelerate capturing and sharing of data? Any bite? Don't be shy. Only 200 people here, and we're recording. That's it. No stress.
I must say, not very much to accelerate capturing and sharing of data. Initiatives such as the OPAL initiative is one way of capturing data through the patient by donation. There's as I explained in my presentation, there is initiatives such as the initiative that we are working on called Medomics, is where we are trying to federate all the data that is existing in different systems in the hospital. But we are in fact at the very early stages of that. In fact, the MUHC is just installing a new office for what's the official name of this position? The position would be information officer. Information officer of the hospital, and they are in the process of working on the digital transformation of the hospital, which includes federating data so that we can access it for clinical use, for research, or for different purposes. But that's just in the initial stages. I would say I'm fairly discouraged in a sense to some extent at the rate at which this process is going. Maybe someone else can add to my comments.
The Montreal Neurological Institute is moving towards open science, and they are trying to move in that direction and definitely are quite committed to it. There are public data sets as part of the NIH and the UK Biobank. There's more and more of these public data sets that are available for research and for those of us working in it, in machine learning. And yeah.
All right.
Yeah, it's a major challenge for what we're looking at, histological images. As I said, there's not a lot of publicly available data sets. Capturing the images is technically challenging, and storing that amount of data is technically challenging. We would like to push for, at least in Montreal and across Quebec, would be having digital pathology becoming sort of the main focus. But then there will be challenges in accessing that information and having patient consent for that as well. One thing we've done is to collaborate with people around the world. So the UK at University of Nottingham, they have a large digital pathology data set. So we've been working with pathologists from around the world to try and access that. But it is, I agree, it is a bit of a frustration in trying to get the data.
Sure, I had a bit to add.
So I think the importance of gathering data is definitely a priority. What is important is that this data is standardized, because if we gather data that is not standardized, we will end by having data that cannot be interpretable. And research needs standardization to start with. So we have to be careful and be clear in our mind that we are talking the same language, because we can gather data and then we find we didn't do it right, and it's a very costly process.
Very good. All right. Next question. Can you elaborate on how you address the mistrust of machine learning and actual ground truth data? It's targeted to you, Tal. So it's she or he is asking, can you elaborate on how mistrust is addressed from mistrust of machine learning versus when it compares to actual ground truth data? How do you address mistrust? That's the question, I guess.
So our group is focusing quite a bit on quantifying the results of predictions. So we're looking at uncertainty and probabilities in our outcomes and providing the clinician with confidence levels for all predictions, whether it's segmentation, so at every pixel, or lesion level, or tumor level, saying I think this is a tumor, I'm not sure, or really even at the prediction level. So I think this patient will progress with a certain amount of confidence. So we're working a lot on those types of measures.
And that was our argument of defining the uncertainty and at least giving a measure instead of having a all black or white answer, right?
That's
so
right.
to appreciate the uncertainty. Yeah. Okay, so how well does the machine learning algorithm compare to a human expert? How did it compare? if you want.
It depends on the task. So again, we are fortunate that the reason I'm working quite a bit in multiple sclerosis, I have a huge amount of data in the tens of thousands. We'll have up to 50,000 MRI, and we work closely with a company who spends a lot of time doing manual segmentation very, very carefully, sometimes several manual raters coming to a consensus on whether something is a lesion or not. So we have really, really good ground truth for segmenting lesions in different contexts. And our algorithms, there is, of course, inter-rater variability and some noise, but our algorithms are working very, very well in comparison with the manual raters. So that's why a lot of these approaches have been integrated into the...
You were careful not to say better than.
No, I would never say better than. The idea is if there's some disagreement among manual raters, it would be an ideal world where we had 10 manual raters doing every single segmentation. We can look at the uncertainty and the variance in the manual rating and see if our approach it comes within that range, but it's so expensive to do in the first place. I don't think we can get 10 or 15 people to do all that segmentation. But that would be the right way to do it.
Yeah, so but it came across in a few presentations that it won't replace, it will more complement, right, and accelerate. That was one of the key points, right?
I think from histological images, especially in breast cancer, there are a number of stages where, as I mentioned, there are these borderlines where there's not a real good agreement between pathologists, to the point where you can have, maybe a 50-50 split on, whether they would call it one stage or the next. So I think, maybe quantifying these is a place that AI can maybe have an impact. There have been some studies to compare on histology, computer vision and machine learning compared to pathologists. So pathologists aren't perfect either. So individuals, so if you have a small number of pathologists, they're not looking. And as you said, you can't have 10 people looking at every single image. So what they have found is that it's a combination that actually performs best. So having a pathologist and the machine working together is actually giving the best results.
Very good. No robots replacement yet for pathologists. As AI research comes, that's a curious one. So as AI research comes from the engineering domain, what challenges arise in successful integration in the medical domain? Are clinicians intimidated by technologists? I told you that'd be tough questions.
My team works very, very closely with the clinicians, with neurologists, with neuroradiologists. We don't work in isolation. We constantly keep the clinical needs in mind, the metrics of success that the clinicians dictate to us in how we determine how our methods are doing. So I guess we are fortunate enough to be all working together to common goals in this particular case that we're looking at.
Absolutely, yeah. Okay, so when can we expect AI-powered tools in the clinic? Can we do as normal citizens? Are there things we can do to speed it up?
So I think they're already, to some extent, in the clinic. For example, auto-segmentation algorithms are already present in some of the software that we have. The optimization algorithms that we use for treatment planning in radiotherapy some of them are taking principles of AI in their functioning. And then going forward, I see that knowledge-based planning is what we call knowledge-based planning is algorithms that learn using regression models from previous planning practice to do new treatment planning. So also there I see commercial products that are entering the clinic, and this will just on the previous question as well. This can never be built into a practice, into a medical practice, if there's not a multidisciplinary team, or if the people with quantitative physical background are not embedded inside a clinical environment. So I think this is extremely important to have multidisciplinary teams working on the integration of AI technology.
Just one additional comment, which I think will probably have a major impact, and it's already happening in some centers, is the adaptive radiotherapy. I showed one slice when the patient anatomy changes during treatment. We treat the patient for 7 weeks on daily basis, 35 treatments, and you can expect that this patient with head and neck lose weight and the tumor shrink, and the anatomy that you are treating at the end of the treatment is not the same anatomy that we started with. So there is a dynamic, continuous change of the patient anatomy, and what we are doing currently is treating them with the same treatment parameters. So we discovered by doing scans that no, the tumor change and the patient change, and we need to adapt the treatment to the patient. But this is extremely difficult process because it has to be done quickly within 24 to 48 hours in order to have any meaningful impact. So the time constraints and the resources that it takes from a department is tremendous. And now this can be done on a daily basis with artificial intelligence, would be a major impact on the outcome of treatment, both on tumor control and side effects. So this I see one of the most important impact of artificial intelligence in head and neck radiotherapy.
All right, so you want to add on?
I want to speak to your previous point, which is a very, very good one. If we want people in computer science, machine learning, and AI to work with real clinical data, again, we need to invest in finding ways to develop federated data sets, well organized and large enough so that those of us working in machine learning could then learn what are some of the challenges that are different in different clinical contexts and really start to try out different and develop new algorithms.
I see.
Yeah.
Another one, actually. So it's more philosophical. You ready? Can we trust predictions made by AI if we don't know what they're based on? How does, speaking to your point, we don't know about those neural networks, what they work. Can you trust them?
But you did speak a little bit to that. So deep learning is the popular machine learning approach, which is leading to many of the advances that we're referring to. And it is technically been a sort of black box. And this is a challenge for the field. There are more and more algorithms that we're all focusing on the context of machine learning within computer vision and medical image analysis to try to open up the black box. And some of it was presented in the context of histology to try to see what was the network looking at when it reached certain conclusions. And I think once we do that, we can then go back to the clinicians and say, hey, actually, we've learned something. The deep learning keeps looking at these areas, and then we can work together to better understand the disease. But that whole area of interpretability is currently an area of interest for the whole community, actually.
Okay, thanks. How can a computer model learn from models of brain scans and when the morphology of a patient with brain abnormality can be vastly different from a normal brain? How does the computer determine patterns in between them? Individual differences versus pathology. Anyone? We've got plenty of questions. Yeah.
Okay, back to me. Big data.
You're doing great.
The answer is big data. The more data we have, the more we can understand what are the patterns of variability. The results that I showed for brain tumors, there was a wide variability, but that was just one type of high-grade and low-grade glioma. That's a public data set that we're all trying our algorithms on. The more data that we have out there, the more we could be variable. One thing that my team is looking at is something called transfer learning. So now that we have huge amounts of data with regards to multiple sclerosis, and we have much less data for certain kinds of brain cancers, can we actually transfer what we learned from MS to diseases, strokes as well, brain diseases that have fewer data sets, and what can we do in that domain?
So we heard about breast cancer, a bit about brain cancer, about head and neck as well. What's the perspective for the other types of cancer? Is there types of cancer, machine learning and image analysis, for example, or other types of analysis like genomics analysis? Is there other types of tumors or sites of cancer that performs better than others using AI methods? Or is it all methods the same?
So in the clinic now, lung cancer is also a very big one that's being looked at. I can quote a couple of studies. One of them is just looking at images of lung scans. The machines are able to know if a lung scan is a lung cancer or a benign disease, at least as well as the radiologists. And in radiation oncology, there's even a study where they try to look at the scan before starting the radiation treatment, and depending on the characteristics of the tumor, again, through AI algorithms, they can try to look at if they can reduce the dose, if the computer can tell the radiation oncologist how to adjust the dose so that we give the least amount of radiation to obtain a good control of the tumor and thus decreasing side effects. So these are two examples in lung cancer.
Yeah, from a general perspective, there's no limit, I think, as to where we can apply these kind of methods. It all hinges on the data. So it comes back to previous questions that have already been asked, but it just hinges on how much data is available for a particular cancer type, what we can expect from the algorithms.
Yeah, I think the AI machine learning is really a toolbox, and many of the algorithms have been around for decades, and they're now being applied and maybe modified. But yeah, I agree. There's no limit to what can be analyzed, and there will be incorporation, as we saw here. Incorporation through multiple different modalities, so incorporating genetic data, maybe blood test data, medical imaging, histology, all of that can potentially have a much richer data set. So right now we're all kind of working in little silos where we're working on different types of data. And there's other groups working at genomics and whatnot, but I think the goal probably would be that we could integrate all of these into a bigger sort of model that would really be better encompass the patient.
Very good. So quick fire questions on the Opal app. Are you guys ready? Can you hear from Tarek? All right. So there's a person here who wants to know, how is the data input into Opal? Does the patient have any work to do to input it, and how much time does it require to input the data?
No, the data comes directly from the electronic medical record. So it's automatic. The patient doesn't have to enter any data to see their data, but we can send questionnaires to the patients about their symptoms, and those data the patient needs to enter.
All right. So can Opal provide reference to patient support groups?
At the moment, we have some support information in there that was provided by the Cancer Mission Patients Committee, but we would like to add in some more, and we will be adding more.
Okay, and more technical, what is Opal available on in terms of platform, cell phone, laptops, etc.?
So right now it's iOS, iPhones, and Android, and we're also working on a web app version.
Okay. And so we have time maybe for two more, right? You guys are doing well? Two more. Okay, one for Dr. Shinoda. How is physiological movements such as breathing accounted during radiation? It's more very technical question, but I'm sure.
Yeah, there is definitely in the head and neck region movement during treatment. The least of them is swallowing. And when we swallow, the larynx moves up about 1 centimeter. So this has to be accounted in the volume that we decide to treat the patient in the head and neck region. And definitely by having observation of the patient during treatment and having large database, we can minimize this additional volume that we add to compensate for the movement to the least amount, and therefore help reduce the toxicity of the treatment.
Right. So thank you. The last question, if that's okay for you, Tao. Is patient data used in predicting evolution of their tumor? I think we've addressed this, but if so, what are the variables? So any role for heuristics? And the question is essentially, what are the variables allowing you to predict the evolution of a tumor, in, let's say, brain tumor?
So the question is, what variables I use, or?
Yeah, so what are the variables that can be used, I think in general, to predict the outcome of the evolution of the tumor?
Oh, like to predict the future?
That's right, progression.
So we're still working on this problem, but the idea is that you use deep learning models, and the deep learning models are figuring out what is interesting and important in the image to actually create, to get the right outcome. So we have training data sets, and you have, binary outcomes, I don't know, progress, non-progress, or new lesions, no new lesions, or whatever it is, or counts, or whatever it is that you want, and you put that into your network. And so you, therefore, you train the network, and then we try to figure out later on, as we open up the black box, what was it looking at when it made those progressions?
What was the computer seeing in it?
Yeah, what was it looking at? But we don't use specific heuristics, and we don't point it at specific areas of the brain.
All right. Well, on this, it's all the time we have, so I think we owe them a big, great round of applause for